Well, we can move on then. So again, if you go to the website for materials, down to compare behavioral state estimates, you can copy the code that I'll be using directly from this file in the GitHub repo. And I'm going to restart my R session. Okay, so I'm using the same packages essentially I used yesterday here that I'm loading in, including all of those different packages for fitting the different methods, such as foie gras, momentum, and Bayes move. So if you had issues with foie gras yesterday, this may or may not work. We're not going to be fitting any models here. We're just going to be like using a function to grab the results that I've saved. So this may or may not work on some people's computers, depending on what they were able to get done yesterday. But at the very least, you'll at least be able to see these comparisons visualized across these methods. All right, so after loading the packages, the first thing I'm going to do is load in some of these model fits from each of these different methods. So since I saved them as, I think it was R data files in my process data folder, yeah, I saved them as R data files. So if you save an R data file, it saves whatever R objects that you had in your environment at that time, which is relatively convenient, especially if you took a bunch of steps to get to that point and you don't think things are in a convenient format to save them in a file type such as a CSV. So things for like a model fits, I can't save that as a CSV file. It's easier to save it as an R data file and then load it in here like so. So I'm going to load in these fits or data sets from each method. And those are now all in my environments. So the first thing I'm going to do, like I mentioned, is to wrangle these model outputs into a format that's usable, that I can combine these results across these different methods. So I'm using the join function from foie gras to join the results from the state space model with the move persistence model based on these 8-hour predictions. For the momentum package, I'm taking this last data frame I ended up creating, which I called DAT3, and then adding a new column called states. In the way that you extract the most probable sequence of states from a hidden Markov model fits from that package is using the Viterbi function, which uses the Viterbi algorithm to decode this likely state sequence. And I'm doing this for the model that I fit for 3 behavioral states using the step length, turning angle, and net square displacements, which I think will make the best comparison against the results from the Bayes move package, so the non-parametric Bayesian methods. And then lastly, assigning this DAT out to objects from Bayes move from the M4 method specifically to this object called Bayes res, which is easier to interpret here. So I know what that actually means. It's a better name. Okay, so now kind of going through different pieces of this. First, let's say we wanted to compare the state-dependent distributions. Well, state-dependent distributions are only estimated for hidden Markov models or the non-parametric Bayesian models. So state space models don't have this because there's no finite or discrete states. So it's not a thing that you would ever need to interpret or look at. But we're going to do so again to cover these 2 different methods that were used. So for the hidden Markov model, I'm going to show these results. Let me rearrange the size of this. So these are our different step length distributions that are continuous, estimated by the hidden Markov model. So we have one for this breeding behavior, where they're staying at the breeding ground at Fernando de Noronha. In blue, we have this other density that's also relatively small, but intermediates of the 3. that's representing foraging behavior at the foraging grounds on the mainland. And then in green here, that's really flat and small at the bottom, represents these long, large step lengths by the migratory behavior. And then for turning angles, we have relatively peaked distribution at negative pi or pi for this orange breeding behavioral states. In blue, the foraging behavioral states, this distribution is relatively flat looking by comparison. And then in green, the turning angle distribution for the migratory states has a slight peak here at 0. So it's moving straight. And then lastly, our distributions for displacements show basically a peak that's almost at 0, essentially 0 for the breeding behavioral states. So it's not leaving the island. And then our other two states, we have these much longer, flatter distributions over this whole area, indicating that they're using other spaces further away from the island. So obviously the migratory phase, they're moving away from the island to some distant location. In the foraging grounds, they arrive and settle at a distant location, which could be 400 kilometers away in some instances, but also 800 kilometers away. But they represent essentially the same type of behavior. Okay, so for comparison, we're going to look at now the state-dependent distributions from the M4 model. And here I'm taking some of these model results and then again having to relabel the names for these different estimated states. And then now plotting these. Okay, so these labels on the rows are a little more difficult to read, but the first row is the breeding encamped behavior. So it's an encamped behavioral state at the breeding grounds. The second row is the ARS, or area restricted search behavior at the breeding ground. Then we have foraging 1 and foraging 2, which are essentially the same, just at different distances away from the island. So these could essentially be lumped together. And then the migratory states, which is that migratory behavior from the island to the mainland. So again, we have displacements that's very low here for encamped or ARS, where again, they're not leaving the breeding ground. For encamped, this is defined by the smallest distribution of step lengths because they're not really moving anywhere. But for ARS, they're moving a little bit more. And then for turning angle, the distribution is relatively flat for the encamped states, but is turning around a lot with some movements moving relatively straight for the ARS state. By comparison, the foraging states have displacement either close to 400 kilometers away or about 700-ish kilometers away. And the step lengths are relatively large, but comparable to the ARS states, depending on which one we look at. And the distributions of the turning angles are similar to that of the encamped state at the breeding ground. And then lastly, on the bottom, the migratory state, we have displacement that's kind of spread out across all these different bins, across all distances, has the largest distribution of step lengths that are very long or fast. And these turning angles are very straight. They're centered here around 0 radians, or 0 degrees. So in general, I would say this does match up relatively closely with the hidden Markov model, except that it does appear to identify 4 states if you lump the 2 foraging states together, where that only identified kind of like the breeding states. So maybe just like the breeding encamped, then the breeding ARS might be a separate state, and then the migratory state as a third one. So it does appear that the M4 method was able to pick up on some other behaviors that may be happening that we weren't considering with the hidden Markov model. Now, if we wanted to visualize time series of these state estimates, this is also including the results from the state space model. We can just use the plot function on this model fit for the state space model. So similar, this is what we just looked at yesterday, but just to refresh, So this is the 8-hour predictions from the correlated random walk. And this denotes these pretty clear phases where they're either resident at the islands or they're migrating. So the gamma parameters increasing to 1, and then it drops back off as they reach these foraging grounds on the mainland. So we have another migratory individual here. Then we have our 2 resident individuals, this individual and the next one. And then the last one also exhibits a migratory phase. For the hidden Markov model to plot a time series that's similar to this, we can use the plot states function for momentum. And I need to increase this panel. Let's try this. Still not big enough. Try it again. Here we go. All right. So for these 3 different data streams and 3 behaviors, we see in this first row is the changes in states over time. So it starts off in state 1 for this first individual. So that's our breeding behavior. Then it enters state 3, that's our migratory behavior. And then finally changes to state 2, which is the foraging behavior. And we see the probability changes over time for each of these different behavioral states, 1, 2, and 3 beneath that. So again, this peak or this spike in the middle of the time series for that migratory state. And these seem to be much cleaner looking or easier to interpret compared to some of the other versions of this hidden Markov model that we looked at yesterday. Then we have this second individual again exhibiting a migratory phase, but there's no switches between the foraging and the breeding behavioral states. It has to go through this migratory state first because of the displacement variable that we're including. Then we have an example of a resident individual where it's entirely in state 1 at the breeding ground. Same thing for this individual. And then we have another individual that actually starts off in breeding, migrates, reaches a foraging ground where it's staying, but then actually decides to, in this case, migrate again or move quickly to a separate foraging patch or foraging region where it eventually stays in this foraging state. Okay. And now for the results from M4. Come on. Problematic. Okay. So like I mentioned yesterday, the M4 method is actually able to estimate the proportion of observations belonging to each state per track segment. So it's not necessarily only going to be potentially in one state or another. There's the possibility it's in multiple states during that segment of time. So we can see for these migratory individuals, there's this spike of yellow at some point in the middle of the track, and that corresponds with this migratory phase. Whereas the beginning is primarily this dark purple, and that represents the breeding encamped phase. Although for these 2 individuals here that migrates, there's also some what is labeled breeding ARS behavior occurring during this period as well. And then after that migratory period, these individuals exhibit a type of foraging, whether they're closer to the islands or if they're further away from the island in this greener color. And then ultimately, these 2 resident individuals always stay in this breeding encamped state, and they just are entirely this one behavioral state. So we can see some more nuance in the results from this method by comparison to the other 2, the state space model and the hidden Markov model. But to look at some spatial comparisons mapped out, again, we're loading in the spatial layer for Brazil, and we're going to focus on a single migratory individual here, 205537. And we're going to look at the results for the state space model. Okay, so green circle is indicating the starting location, the red triangle is the ending location, and the colors are indicating for each point our behavioral metric or index for gamma. So the darker they are, the more likely it's to be like a foraging ARS state where it's not moving much and it's moving, it's in a tortuous manner. Whereas the yellower it is, the more likely it's moving quicker and in a straight direction. So if I zoom in at the island to start off, these are all dark color because again, it's not really going anywhere and it's turning around a lot. And as we zoom out, this one, the migratory section of this track is primarily yellow because it's transiting in this case. But once we reach the mainland, it appears to briefly slow down, but then pick up speed a little bit until it reaches this separate foraging area a little bit south of its initial stop. And we can see that it returns to this slower, more tortuous behavioral state again. And for looking at the results from the hidden Markov model, it looks relatively similar, but here now, instead of a continuous value or metric, now we have discrete behaviors. So we have the breeding and camped behavior, the foraging and migratory. And if I zoom in on the islands, this also appears to do a very similar job in identifying these as a slow, tortuous behavior that we identify here as breeding and camped. Everything else here along the migratory track is just labeled migratory. So by comparison, there's no nuance in the directional persistence of these points, which is especially notable as it's reaching the mainland, where it did appear that it was slowing down and potentially changing its behavior slightly. These are all still considered migratory for our purposes here for the hidden Markov model. And then once it reaches this final foraging area is where this behavior switches to a foraging state. So we do see that there's this slight difference in how this model is producing results versus the state space model. And then lastly, for M4, we can make the same type of map. And the results, again, resemble those previous methods. So these these points around the island are again labeled breeding and camped. And then as we move away into this migratory phase of the movements, these are all labeled migratory points. And then at this final foraging areas where they're finally labeled as being in a foraging state. So this is almost identical to the hidden Markov model results, at least for this particular individual and the way it was analyzed. But one thing that's nice that you can do with this data set as well is you can look at the probability of being in one behavior or another. So in this particular behavior, this is for the foraging states. So what's the probability of being in a foraging state? If we look at the beginning of the track, the probability is very low, dark purple. During the migratory phase, it's also quite low. And it's not until you get to ultimately this foraging ground that it goes up to essentially close to 1, about 99% here. And this is due to that segmentation process where it probably identified the break in this track at this point between these two locations. So everything towards the end of the track after that break point was labeled a high probability of being in a foraging state. All right, so that does it for the code for this portion that I wasn't able to cover yesterday. Obviously, this is just kind of a brief snapshot of this comparison. You would obviously potentially want to do this for multiple individuals and maybe with different time intervals of interest, depending on what inferences you wanted to make based on these data sets. But I think this is a nice introduction to making these comparisons as to how these different methods can be used.